So the last but not least, I'd like to introduce the last speaker, and thanks to everyone for staying here. Right. So Dr. Lin is a physician scientist, received all her training and working at National Taiwan University. And he's a medical oncologist and also do early drug development, early phase clinical trials. And so that's my colleague, and I'd like to hear what's the clinical development of KRAS target therapy. Thank you.
Thank you. Thank you for the organizing committee to invite me to talk the last talk today. So this is between now and dinner. So I will do it very quick. So my topic is Ras drug. So basically, is derived from the Kevin Shortcack recent book. They published a book called the Ras drug discovery. So I changed the title to the KRAS drug clinical development. Here's my disclosure. So actually, I did and I'm doing clinical trials with a lot of pharmaceutical companies, including the companies I will mention later in my talk. So the most prevalent KRAS-driven cancers are lung cancer, colon cancer, and pancreatic cancer. And while some other cancer like melanoma tend to use NRAS rather than KRAS, or the bladder cancer tend to use HRAS rather than KRAS. Actually, I think currently scientists still don't have the clear answer. But the KRAS are prevalent in certain in these three types of cancer. But if you look into the detail, actually 19% of KRAS mutation in pancreatic cancer are G12, and the 68% of colon cancer mutation in KRAS mutation is also G12. But lung cancer G12 mutation, G12X mutation, is 85%. And if we look into the G12 mutation of these three types of cancer, actually the most prevalent for the pancreatic cancer is G12D. The most prevalent for the colon cancer also G12D, and most prevalent in the lung cancer KRAS mutation G12 is G12C. So the different mutation probably can drive the tumor types, but what they play in the biology or in the therapeutic, what do they mean in the therapeutic area? I think there's still much more about to learn. Let's use the lung cancer, the G12C as an example. So it's a transfer mutation, the glycine switch to cysteine, and more common in not only in the non-smoker, but also common in smokers. And about 60% of KRAS G12C mutation are in women, and 95%, almost all are adenocarcinoma, but not other tumor types. And in lung cancer, unlike other driver mutations, it usually associated with tumor immune alteration. So it's associated with a higher tumor PD-L1 expression, and also higher associated with higher tumor mutational burden. In the American studies, actually G12C mutation, KRAS G12C mutation in lung cancer actually are present in with ethnicity difference. So the Caucasian has a 15% prevalence. African American has a 12% prevalence, and the East Asian Americans actually has only have 4% prevalence. And why there's a difference, and still no answer today. Skip this one. Targeting KRAS has a long history. Actually, in the clinical development, the formal phase 3 clinical trials have been conducted using other approaches other than direct targeting KRAS. For example, this is a phase 3 clinical trial targeting MEK. MEK is a molecule one downstream of KRAS. So they're using the MEK inhibitor selumetinib plus chemotherapy, microtubule interactive agents, docetaxel, versus docetaxel alone as a so-called standard therapy in the non-small cell lung cancer, stage 4 non-small cell lung cancer with a KRAS mutant, and that it did not show any benefit to prolong the patient's survival. And there's a lot of explanation about this, why we target the KRAS downstream signals that cannot help the chemotherapy to improve its efficacy. So there might be insufficient target inhibition, and or this particular MEK inhibitor probably lack a therapeutic window, so not a very good therapeutic window, or the MAPK pathway, PI3K pathway has a crosstalk feedback adaptation, and so on, or there's because of tumor heterogeneities. So Dr. Chia already mentioned that in the 2013, Dr. Kevin Shortcut found that there's a new pocket in the KRAS, so-called switch II region that could develop the inhibitors, and the first targeting the G12C because they use a cysteine disulfide bond to inhibit the switch II regions, and they quickly developed in vitro inhibitors, in vitro inhibitor in 2016, and they developed an in vivo inhibitor in 2018. And then there were two compounds enter into the clinical trials roughly in the same period. The first one, sotorasib, that's followed by the adagrasib in 2019. So from the very first the discovery to the first clinical trial, it took 6 years. And then after first clinical trial, actually got US FDA approval quite quickly, only 2 to 3 years. So the G12C actually, the first clinical G12C inhibitor actually targeting the off state, so the KRAS GDP binding state. They will lock the KRAS in the GDP binding state and will not go to the GTP binding state, and then the tumor will stop growing and have a therapeutic effect. So the first phase 1 trial for the sotorasib, then called the AMG 510, so the Amgen's by Amgen. So it's a Cobra 100. Cobra 100 is actually is a complicated study, but one arm in that trial is non-small cell lung cancer arms. So they used a 960 milligram dose of sotorasib to treat a group of people, a group of more than 126 non-small cell lung cancer patients, and among them 46 patients that responded to the G12C inhibitor sotorasib. So the response rate is 37%, so it's roughly 40%. And the median progression-free survival is 6 to 7 months, and the median overall survival is double, so it's a 12 to 13 months. And the patient, in terms of the adverse events, side effects, there's a severe or life-threatening grade 3 or 4, the treatment-related adverse events occur in 20% of patients. And the sotorasib later moved to the randomized phase 3 trial, try to determine its role in the non-small cell lung cancer. So it's a CodeBreak 200. CodeBreak 200 phase 3 trial was first presented by presented in ESMO 2022, and later published a full paper in the Lancet 2023. So in this phase 3 trial, they compared the sotorasib with chemotherapy, docetaxel, microtubule interactive agents. And the primary endpoint for the trial is progression-free survival by independent review. So the progression-free survival meaning the patient received the treatment sotorasib or docetaxel when the patient first hit the progression or the patient passed away, whichever comes first. They're called the progression-free survival. And it showed the progression-free survival benefit. So the hazard ratio was 6.66, and the median survival 50%. Median survival improved from 5.6 months to 4.5 months to 5.6 months. Probably rough here, but if you look at the patient at risk, actually there's some criticism for these trials. So for the control arm, docetaxel arm, a lot of patients have been censored pretty early on. So the difference in the later part of the survival curves could not be interpreted very properly. Adagrasib is another KRAS G12C inhibitor developed by Mirati. So it's an MRTX849. The first phase 1 trial called the Crystal 1, there was a non-small cell lung cancer arm. So they treated 112 patients. Among them, 48 patients responded to the adagrasib. So the response rate improved from, but might be better than sotorasib. So it's more than 40%. Sotorasib is less than 40%. Progression-free survival, again, between 6 and 7 months for the advanced non-small cell lung cancer, and the median survival is double, is between 12 and 13 months. And treatment-related adverse events, great severe, life-threatening, and even 2 as causing to a patient pass away. The percentage is 45%. The adagrasib also did a very similar phase 3 study called the Crystal 12 in non-small cell lung cancer. So it's a randomized phase 3 trial compare adagrasib to docetaxel, but it's randomized to 2 to 1. So two-thirds of patients received adagrasib, 1/3 of patients were allocated to docetaxel. The median primary endpoint is also the progression-free survival, improved from 3.8 months to 5.5 months. So hazard ratio is 0.58. So it's a clinically is a statistical significance. So in it was first presented in ASCO 2024, later published paper in the Lancet 2025. If you look at the Lancet 2025 papers, the patient at risk, there's another row of numbers putting the patient's sensors because they want to correct the problem of the sotorasib phase 3 trial. So let's do the cross-trial comparison, comparing the different safety profile between these 2 inhibitors, because these 2 are the only 2 inhibitors so far have get FDA approval. If both compare with chemotherapy, basically chemotherapy had more stomatitis and bone marrow suppression. I think that is quite obvious. But both sotorasib and adagrasib have more GI side effects. For example, diarrhea, AST, ALT elevation compared to the chemotherapy. And for sotorasib, most of the AST, ALT elevation actually is grade 3 or 4, so it means severe or life-threatening. Definitely need to stop the medication, and sometimes need the dose modification or the drug discontinuation. And adagrasib pretty much show the same things. So there are some benefit over, there are some good part over chemotherapy, but they have their own problems. And in terms of efficacy, adagrasib, both sotorasib and adagrasib show activity in the intracranial metastasis because of brain had a BBB, so the small molecule might not be able to close a BBB to target the brain metastasis. Sotorasib only had case reports, case series. Adagrasib systemically evaluated this question in their phase 3 trials. So in their phase 3 trial, part of patient had a brain metastasis, and actually adagrasib actually shows activity in terms of the shrinking the tumor in the brain or prolonging the progression in the brain. And then because G12C, the second, colon cancer also had prevalent G12C mutations. So both sotorasib and adagrasib also had clinical development in the colorectal cancer, KRAS G12C mutation. But again, this time, both agents take similar approach. They are not developing this agent as a single agent in the colon cancer. And both of them combine their agent with anti-EGFR antibody. For example, sotorasib, KRAS G12C inhibitor combined with panitumumab, anti-EGFR antibody. So they have use this combination to treat patients in the colorectal cancer with a response rate 30%, median progression-free survival 5.7 months, and overall survival 15.12 months. And if you look at the treatment-related grade 3 or 4 adverse events, it's a 27%. So compared with the only single agent sotorasib, it's not significantly higher. So you use combination is feasible and had a reasonable response rate. And the sotorasib alone to treat colorectal cancer G12C probably only had a 7% response rate in the phase 1 trial. So they did a phase 3 trial, CoBRA 300 in the colorectal cancer. So comparing sotorasib plus panitumumab with standard of care in the colorectal cancer. So standard of care in the colorectal cancer in this setting is using chemotherapy, chemotherapy called trifluridine tipiracil, or targeted therapy called regorafenib. So in this trial, actually sotorasib plus panitumumab definitely improved the progression-free survival compared with standard, so-called standard therapies. But the overall survival did not improve, did not seem to improve in the trials. Undruggable also did a phase 1 trial combined with cetuximab in the colorectal cancer at a 20% response rate, 5.6% median progression-free survival, and the treatment-related adverse events also not increased compared with a single agent, not increased too much compared with a single agent. And undruggable plus cetuximab, they had already done the phase 3 trial, the so-called Crystal 10 in the colorectal cancer G12C mutations, but the results are still under analysis and not reported yet. So we will see the report probably next year. And okay. So actually there are other companies also developing G12C inhibitors in this area, in the non-small cell lung cancer, colorectal cancer. Usually we can call it the second generation G12C inhibitors, but they are really better tolerated, more efficacious, probably not, because if we see the Roche compound, divarasib, response rate in lung cancer is 55%. And Novartis compound, undruggable, response rate in lung cancer is 45%, very small study. And the Eli Lilly compound, olomorasib, the response rate is 35% in the non-colorectal cancer. And Merck compound, calderasib, the small cell lung cancer cohort, the response rate is 42%. So probably not too much efficacious, but maybe more better tolerated. So it better tolerated, more combinable with other agents, and we will see. Resistance mechanism actually developed because the progression-free survival is not forever. So patients actually did progress, or so there's a definite resistance mechanism. Resistance mechanism came out in roughly in the 2021 with 3 papers in the New England Journal of Medicine, Cancer Discovery, and Nature. For example, in the New England Journal of Medicine, they analyzed 17 patients receiving undruggable, find that various different mechanisms of action. Some actually developed other KRAS mutations, and some others had developed KRAS amplifications, and some developed other signal pathways mutations, like downstream signal MAPK pathway, even PI3K pathway, and some actually had a gene fusion as a resistant mechanism. Divarasib also did a similar thing in the clinical trials among the 16 patients in their clinical trials. Also find various resistant mechanisms. So they did have resistant mechanism, but there's no predominant resistant mechanism. We can just moving forward very straightforward. So the way to overcome this is to target targeting other targets in the RAS signaling pathway. For example, targeting KRAS on state, or targeting SAS1, or even targeting SHIP2. And I will show you some example. One is to targeting on state, so it's RMC 6291. So elironrasib. Elironrasib is using the tricomplex to lock the RAS in the on state and then not go to the off state. So inhibit the tumor. Dr. Xie already mentioned this mechanism, published in Science. This is a science published by Revolution Medicine scientists. I think it's James Christensen. And the clinical trial report result, phase 1, have been recently reported in Boston triple meeting. So in the phase 1 trial, if the patient had received the other G12C inhibitor, non-small cell lung cancer, receiving the off-state G12C, so had received off-state inhibitor, and then received this on-state inhibitor. Although patient number are very small, but they have 42% patients responded to these agents. And how about the patient not receiving any G12C inhibitor to receive the G12C on inhibitor from the start as a first targeted therapy? The data will be reported in the ESMO Asia in Singapore 3 weeks from now. So you will see that. Combining, I'll just use one combination, and then I will start my talk. So the combination have been tried in clinical trials. The most advanced is the SHP2 inhibitors. So there's a lot of SHP2 inhibitors combining with the different KRAS G12C inhibitors. If you ask me why this combines with this usually they are from the same company or they collaborate with one company. So that's the reason. So there's a not scientific reason between why this should be combined with this one. So for example, the Chinese company, they use their SHP2 inhibitor JAP3312 combined with their own G12C inhibitor, glycyrrhizib. This has been approved in China. This compound has been approved in China. So they reported the results in the last year at ASCO, showed that this is a phase 1 result. The response was quite promising. So the 70s, although all the patients did not receive the KRAS G12C inhibitor before, so it's a G12C inhibitor naive, but 77% of patients responding to this combination. If we remember the sotorasib and adagrasib only have 30-40% response. This combination seems to be very promising, and more importantly, the adverse events did not increase too much. So come to a conclusion. KRAS G12C mutation is the most common in the non-small cell lung cancer, colorectal cancer, and a few pancreatic cancer patients have these mutations. And the sotorasib and adagrasib are effective treatment for the KRAS G12C mutant non-small cell lung cancer, and for colorectal cancer need to combine with anti-EGFR antibodies. And various adaptive resistance mechanisms have been elucidated in patients, not just in animal model, in patients. So combination with the other RAS pathway inhibitors might be the next way to move forward. Thank you very much for your attention.
Thank you very much. Any questions? There's a lot of discussion about the KRAS target therapy this afternoon. If not, maybe I'll ask a question. So Dr. Lin, how do you see this, the KRAS target therapy currently is the second line standard of care, right? Do you see that it's going to move to the first line therapy?
Yes. So that for the lung cancer, actually there are clinical trials using KRAS G12C in the first line, but usually it's combining with the other standard agent in the lung cancer. For example, chemotherapy or with the immunotherapy. Yeah.
Great. I think we see that this thing, right? There's actually the KRAS target therapy across multiple tumor type. That's a new thing. We're talking about tumor agnostic treatment, but each of tumor biology is actually quite different between lung cancer and colorectal cancer and pancreatic cancer, right? That's great. Yeah. Any other questions? With now, we probably just thank you very much, Dr. Lin, and we close this section. So I think at the end, whether I would just like invite the director, Dr. Sutu, to closing the meeting. Thank you very much.